Okay, we are starting with slide number 27 of the chapter 17 slides, and we are covering the process of transcription, which is the first step in the production of a protein. So during transcription, we know that transcription happens in the nucleus of eukaryotic cells. So during transcription, one of the enzymes that is going to be used or be necessary for transcription is RNA polymerase. And this makes sense because remember in DNA replication that it was DNA polymerase that added nucleotides to the old strand to form the new strand. So now it's going to be RNA polymerase that adds the RNA nucleotides to form the RNA strand. Okay? So RNA synthesis, in other words, we're making messenger RNA, so that's RNA synthesis, is catalyzed by RNA polymerase, which pries the DNA strands apart. So we don't use a helicase in this process. RNA polymerase plays the role of helicase as well. It's going to unwind and unzip the DNA strands and then join the RNA nucleotides to the DNA template strand. So the RNA is going to be complementary to the DNA template strand. And RNA polymerase does not need a primer, so there's no primer. I'm not going to highlight that, but all you have is RNA polymerase, which unwinds and unzips the DNA strand and joins the nucleotides to the template strand. And then the base pairing rules are going to be the same as DNA, except that uracil is in place of thymine. You're going to have U everywhere there would be a T if it was DNA. So RNA has U instead of T. And this shows you transcription. There is a promoter region of the DNA, and that's where the RNA polymerase is gonna attach. And then it's going to come along and unwind the DNA and add nucleotides at the same time. Okay, so those nucleotides will form the messenger RNA transcript, and it will be over when the RNA polymerase reaches a stop codon. And there's an animation on transcription, little video animation, slide number 29. All right, the promoter is defined as the DNA sequence where the RNA polymerase attaches. And then the terminator is the DNA, well, that's bacteria. Okay. So I'm not going to worry about teaching about bacteria. So in the eukaryotic cell, there are these additional proteins called transcription factors, and they help guide the RNA polymerase to bind and begin transcription, initiate transcription. And the promoter element in eukaryotes is called a Tata box. Don't you love that? So the promoter element in the DNA, in other words, the region of the DNA where transcription begins, is called a Tata box because it's like a sequence of thymine, adenine, thymine, adenine. So here's an example of a Tata box, okay? So a Tata box is the promoter region of the DNA where this purple molecule is RNA polymerase. So RNA polymerase will bind to the Tata box along with the transcription factors, and they will all move along the DNA, unwinding and unzipping it and adding the nucleotides. and this is just a bigger picture so you can see the process a little bit better. We started with the nucleotides that have already been made are down here. So the next nucleotide to be added is going to be a U, because this would be, the A would be complementary to the T, U would be complementary to this A on the template DNA, and then this U will be added to this A. Okay, so that's pretty much how that works, and the molecule that does this, that adds those nucleotides to form the messenger RNA, the enzyme is called RNA polymerase that does that. Okay, once the pre-messenger RNA is has been completed, that is the messenger RNA that is produced only from the RNA polymerase, okay? After that, it is processed. So RNA processing is what we're going to talk about next. What happens to that pre-messenger RNA before it is formed into the final messenger RNA molecule, okay? So one thing that happens is, let's see, a cap and a tail are added, and then you have splicing. Yeah, okay, they're doing it. I was just wondering about the order. All right, so the 5' end of the messenger, the pre-messenger RNA, or the beginning of it, receives a 5' cap, and it's the cap is modified guanine nucleotide. It's actually a G, a modified G. So there's a cap, which is a modified guanine that's added to one end, and then many adenines, many A's, we call it a poly-A tail, is added to the other end, the 3' end. So one end gets a cap, the other end gets a tail. Then we have something called, and here's what it looks like. The guanine is modified. It's got 3 phosphates. So guanine is added at the beginning, that's the cap, and then many adenines form the poly-A tail, all right? Then we have RNA splicing. There are regions of the messenger RNA that code, that are called coding regions that are expressed. Then there are regions that are not expressed. They're non-coding segments. The non-coding segments are called introns, and the introns are spliced out. They're removed, and then the pieces of pre-messenger RNA are spliced back together, basically. So exons are expressed. That's how you remember it. Exon starts with EX. Exons are expressed. So these remain in the final messenger RNA, and they code for amino acids. So this is the difference in the pre-messenger RNA that has a cap and a tail added to it. The next step is the introns are removed, and all of these exons are spliced together. We call that RNA splicing. And the spliceosomes remove the introns. So they're called spliceosomes that remove the introns. And then they also splice the exons together. Here's an overview of transcription in bacteria, so don't watch it. Ribozymes can splice RNA. So ribozymes can actually splice or glue the exons back together. All right. So ribozymes are actually made of RNA, and they can splice the pre-mRNA exons together and form the final messenger RNA. Okay? So there is something that I want to mention to you. It's called alternative RNA splicing, and that is So some genes, some pieces of DNA that code for a polypeptide can actually code for more than one kind of polypeptide, depending on which segments are removed during the splicing, or which are left in, which exons are left in. So if certain exons are left in, then you would get one protein. And if different exons are left in, you would get a different protein. And that is called alternative RNA splicing. All right. So this shows you how that works. We've got 3 exons here, exon 1, 2, and 3. So depending on which ones are left in, that would give you like a different protein. Okay, now we're going to talk about translation, and translation happens outside the nucleus. So we've got our final messenger RNA that has been produced. RNA processing has occurred, and the final messenger RNA molecule has been produced, and now it leaves the nucleus, and translation is going to occur. So the messenger RNA is translated into a protein, but there's another type of RNA that helps, and it's called tRNA or transfer RNA. So each tRNA only has 3 nucleotides, and those 3 nucleotides are called anticodons. They're complementary to one of the codons on the messenger RNA. And each transfer RNA transfers amino acids to the growing polypeptide. In other words, the tRNAs transfer amino acids to the ribosome, and those amino acids are added to the polypeptide chain so that it gets longer. Okay? And this is how it looks. So is this a picture. Okay, so looking at this picture, we have the messenger RNA molecule, and it's showing 3 different codons. There's UGG, UUU, and GGC. These codons code for specific amino acids, of course. And so the messenger RNA has left the nucleus, and now it has attached to a ribosome. This brown molecule is the ribosome. So messenger RNA is attached to the ribosome, and the first position on the ribosome, it's not showing you. Okay, so we'll get to that later. So notice that each codon, so this codon here, UUU, the complementary anticodon on the transfer RNA would be AAA. So the transfer RNA that has the anticodon that's complementary to UUU will bring its amino acid and add it to the polypeptide. In this case, phenylalanine is coded for by UUU. So this transfer RNA brings phenylalanine to the polypeptide, and the polypeptide just keeps growing. This next one comes in and brings glycine, because glycine is coded for by the codon GGC, and the transfer RNA that carries glycine to the messenger RNA has an anticodon that's complementary to GGC. The anticodon is CCG. Okay, so that's how it works. That's how the correct amino acids get added, so that they're not out of order. All right, so each transfer RNA has 2 parts. It has an anticodon that is complementary to the codon on the messenger RNA, and it has an amino acid, whichever amino acid is coded for by the messenger RNA codon, okay, that it pairs with. So a transfer RNA has an amino acid and an anticodon. All right. And then this part, I'm not going to highlight anything, but ribosomal RNA is the third type of RNA. The ribosomal RNAs are the ones that actually compose the ribosome, and the ribosome is composed of a large and a small subunit. Now, we do need to talk about the 3 different sites on the ribosome for binding of transfer RNA. There's a P site. The P site is where the polypeptide chain is growing. So it represents the growing polypeptide chain. The A site, I always think of P as standing for the word permanent because it's when the amino acid that's been added becomes a permanent part of the polypeptide. And the A site stands for amino acid, but I always think of it as meaning added, because this is the site where the next amino acid is added to the polypeptide chain. So after it's added, then it moves to the P site where it becomes a permanent part of the polypeptide chain. And E for exit. The E site is where the transfer RNA will actually leave or exit the ribosome. So when we look at this, your messenger RNA strand just keeps moving. It keeps scooting basically to the left, okay? So if we look at this picture, the E site is where the transfer RNA leaves. This transfer RNA is exiting because it has already brought in its amino acid. The amino acid has been permanently added to the polypeptide, and there's no need for this transfer RNA anymore, so it leaves. That's the E site. And then this site here is the P site. It's EPA, Environmental Protection Agency. So the P site is where the amino acid that was added previously in the A site, it's now attached to the other amino acids. It is a permanent part of the polypeptide chain. Then in the A site, which is the first place that transfer RNA will bind, that is where transfer RNA will come in and attach, is the A site or added site. So basically it goes A, P, E is the order. Transfer RNA comes in and adds the amino acid at the A site. Then it moves into, the messenger RNA moves down, and that transfer RNA is now in the P site, and the amino acid joins the polypeptide. Then the messenger RNA scoots more to the left, or really, I guess it would, yes, it scoots to the left. And then that transfer RNA is now in the E or exit site, and it leaves because it's no longer needed. So some things to know is the start codon in any polypeptide is always AUG, and it codes for the amino acid methionine. All right, so that is always the first codon on any messenger RNA, and methionine is always the first amino acid. MET or methionine is always right here. MET is always the first amino acid in any protein. All right. And here is just a picture that shows you the process, starting with step 1. The transfer RNA is going to come in here to the A site and add its amino acid. Then it will move to the P site where the amino acid will be attached. So it will leave the A site, scoot over into the P site. And it's not transfer RNA that's moving, it's the messenger RNA. So the messenger RNA will move to the left, and now what was in the A site will be in the P site. So once that happens, the amino acid that it brought in is going to be added to the chain, or it's going to be a permanent part of the chain. So it'll move over here. Here's when it moves, okay? Then the final thing is that this transfer RNA, the messenger RNA will move, so this transfer RNA will be in the E site and it will leave. So again, transfer RNA comes into the A site or added site. Then it moves to the P site once the amino acid has joined the polypeptide. And then it moves into the E site and it leaves. All right, so the start codon is always AUG. The stop codons can be UAG, UAA, or UGA. But once a stop codon is reached on the messenger RNA, then you no longer, that's when the process of translation is complete and your polypeptide is made. Now, it can be modified, and it is modified because we know that proteins have a complex three-dimensional shape. So after the polypeptide is made, then it is folded either into an alpha helix or a beta pleated sheet, and then its tertiary structure, it's formed into its tertiary structure, which is its final three-dimensional shape. So the polypeptide will undergo shape changes, complex shape changes, and structure changes before okay, here's a good animation overview of protein synthesis, but I don't see the link. If the link is there, it's on slide 83, but for me, I'm not seeing it. Okay, when we come back, I've gone a little bit over for my recordings. I try to keep them 20 minutes or less, but anyway, so when I come back, the next recording, we're going to talk about mutations.